This protocol is designed to rapidly isolate cells from screening interfaces for genomic characterization, and this capability is significant because it can combine microscopically observable cell features with genomic information. The targeted bacteria cells from the screening interfaces can be isolated with high spatial precision in an operationally simple manner using this technique. The hydrogels can be implemented in other screening interfaces. The hydrogel material can be readily incorporated into microfluidic channels for on-demand retrieval of cells from microfluidic devices.
Begin by inoculating the cross-linking buffer with the desired cell density. Prepare the hydrogel precursor solution in a 0.5 milliliter microcentrifuge tube by adding 12.5 microliters of the cross-linking buffer and 5.6 microliters of PEG-ONB diacrylate solution. And lastly, add 6.9 microliters of 4-arm PEG-thiol solution to the mixture. For cell encapsulation in the hydrogel precursor solution, place the thiolated base cover slips on a clean petri dish and position 2 spacers on the 2 opposing sides of the cover slip. Fix the spacers on the base cover slip by taping the spacers to the petri dish. After adding the desired volume of the precursor solution on a non-reactive perfluoroalkylated glass slide, place the slide on the base cover slip and allow the hydrogel formation to complete at room temperature for 25 minutes. Once the gelation is complete, gently remove the perfluoroalkylated glass slide. Place the substrate in a 60 by 15 mm petri dish containing ATGN media supplemented with antibiotic.